Welcome back to another Funakoshi Company technical webinar. In this presentation, I'll introduce you an innovative new research tool, FaoBlue. Our FaoBlue is the world's first reagent for detecting fatty acid beta-oxidation activity directly in live cells. Our presentation is divided into three sections. First, I'll provide some background about FaoBlue. Second, I'll discuss about theory and features. Finally, we'll move on to the FaoBlue applications. I'd like to begin discussion about FaoBlue background. Fatty acids play a key role as basic building blocks and precursors for various kinds of lipids. Inside cells, fatty acids with 16 or 18 carbon atoms are mostly present, and these fatty acids are converted to acyl-CoA forms. These acyl-CoAs are used for making phospholipids and glycerides. Phospholipids are main structural components of cell membrane. Glycerides are stored as energy source in fat tissues. On the other hand, fatty acids are carried into the mitochondria and broken down by beta-oxidation and TCA cycle to produce ATP for energy use. Please note that beta-oxidation is a metabolic process involving multiple steps by which fatty acid molecules are broken down to produce energy. Here's an overview of fatty acid beta-oxidation, or FAO cycle, in mitochondria. Fatty acids are mainly degraded by fatty acid beta-oxidation pathway. The carnitine shuttle is responsible for importing fatty acids into the mitochondria. Once inside the mitochondria, beta-oxidation continues until the acyl-CoA chain has been completely broken down. To put it simply, fatty acid beta-oxidation is a reaction for shortening fatty acid side chains during every cycle. The length of fatty acids becomes shorter by 2 carbons each by the FAO pathway. As you can see, FAO is a complicated biochemical event associated with many types of enzymes, so it is impossible to reconstruct FAO cycle in vitro assay. Also, partial analyses do not reflect the entire FAO activity. Again, it's difficult to evaluate beta-oxidation activity because many enzymes are involved. As for the conventional methods to evaluate FAO activity, there are only a few indirect methods, such as using isotope and analyzing expression level, or measuring oxygen consumption, are commonly performed. For the isotope method, beta-oxidation metabolites are tracked using carbon-13 labeled fatty acids. Advantage of this method is intermediates can be quantified accurately. Disadvantage is costly, radioactive waste is created and expert techniques are required. For the expression level analysis, genes or proteins related to beta-oxidation are analyzed and the activity of beta-oxidation is estimated. Advantage of this method is researchers can experiment with only basic techniques. Disadvantage is it's unclear if the expression level is the same as the activity because beta-oxidation is a multi-stage reaction involved with many factors. The other conventional method is to measure oxygen consumption originated from FAO using phosphorescent oxygen sensing probe. However, this method does not monitor FAO activity directly. As you know, oxygen consumption occurs ubiquitously in cells, so this method measures not only FAO activity but also total oxygen consumption. The main point I'd like to say in this section is to analyze beta-oxidation, a simple, accurate, low cost, and high throughput methods are required. In this section, I'm going to talk about the theory and features. FaoBlue is a prodrug type probe mimicking fatty acids. It consists of coumarin derivative part, non-anoic acid part, and acetyl-methyl group as the prodrug protecting group. Acetyl-methyl group increases cell permeability, allowing FaoBlue to permeate into cells through direct penetration of cell membrane, and its acetyl-methyl ester is hydrolyzed by intracellular esterases. Once inside the cells, FaoBlue behaves just like any fatty acid and are broken down by beta-oxidation. Please also remember, fatty acid labeled with coumarin derivative is a blue fluorescent dye and will not fluoresce at 405 nanometer excitation. This slide shows fluorescence theory of FaoBlue detection reagent. As I discussed in the previous slide, FaoBlue enters into cells and after hydrolysis it becomes free fatty acid type of FaoBlue. The free fatty acid type of FaoBlue is converted into acyl-CoA type, and the acyl-CoA type or activated FaoBlue is transported into the mitochondria and incorporated into the FAO pathway. In detail, acyl-CoA type of FaoBlue or activated FaoBlue is transported into the mitochondria matrix. Activated FaoBlue is degraded by 3 FAO cycles. After the 4th FAO degradation, coumarin dye is finally released and spreads throughout the cell. This coumarin shows strong blue fluorescence excited by 405 nanometers. Please note, FaoBlue shows no fluorescence excited by 405 nanometers before metabolizing by FAO. However, the released coumarin derived from the FAO cycles shows strong blue fluorescence excited by 405 nanometers. In other words, FaoBlue passing through the beta-oxidation in a total of 4 times, coumarins are released and emit strong blue fluorescence. This means the blue fluorescence intensity is dependent on beta-oxidation activity. This slide is the absorbance and fluorescence spectra of FaoBlue and the coumarin derivative dye released after metabolization of FAO. If you observe the left chart, an absorption peak of coumarin derivative as indicated by the red line is different from the FaoBlue as indicated by the blue line by passing through beta oxidation. If you observe the right chart, the fluorescence spectra when excited at 405 nanometer, coumarin derivative dye as indicated by the red line shows strong blue fluorescence, but FaoBlue as indicated by the blue line hardly fluoresces. Please look at gray dashed line from the right chart. When FaoBlue is excited at 350 nanometers, there is zero fluorescence. Please keep in mind, FaoBlue before metabolization shows strong violet fluorescence when excited at 350 nanometers. FaoBlue protocol is simple. Just add FaoBlue to cell culture medium. First, add FaoBlue in fresh Hepes buffered saline. Second, remove culture medium and wash cells. Third, add the FaoBlue solution to the cells. Fourth, incubate the cells over 30 minutes. Finally, you can observe the blue fluorescence by microscope. Please note, we recommend to use FaoBlue with nutrient-free Hepes buffered saline and serum-free medium. As you might notice, FAO reaction normally occurs when there is no external nutrients. This means FAO reaction hardly proceeds under enriched conditions like nutrient medium containing glucose. Thus, nutrient-free Hepes buffer is highly recommended. Moreover, there is a possibility that FaoBlue adheres to FBS proteins. Serum-free medium is highly recommended. Let's move on to the applications using FaoBlue. Here is fluorescence imaging of FAO activity in 4 cancer cell lines. Each cell line was treated with FaoBlue with or without pretreatment of potent FAO inhibitor etomoxor, and fluorescence was observed. All cell lines exhibited blue fluorescence. However, pretreatment of FAO inhibitor clearly decreased fluorescence. These results indicate the blue fluorescence was derived from FAO activity. Like this slide, FaoBlue can be used to compare beta-oxidation activity among different cell types. Here is fluorescence image of FAO activity in HepG2 cells upon treatment with 2 different chemicals. HepG2 cells were pre-incubated with an activator of lipid metabolism or partial FAO inhibitor. After drug treatment, the cells were incubated with FaoBlue for 30 minutes. Compared with the control cells, pretreatment with an activator significantly increased blue fluorescence. On the other hand, pretreatment with FAO inhibitor clearly decreased blue fluorescence. Like this slide, FaoBlue can be used to quantify FAO activity under drug or genetic stimulation. This slide shows a change of FAO activity by ND630, a drug candidate for NAFLD, non-alcoholic fatty liver disease. NAFLD is an umbrella term for a range of liver conditions affecting people who drink little alcohol. As the name implies, the main characteristic of NAFLD is too much fat stored in liver cells. ND630 is an inhibitor of acetyl-CoA carboxylase and considered as a potential therapeutic drug for NAFLD. HepG2 cells were pre-incubated with various concentrations of ND630 followed with FaoBlue for 30 minutes, and blue fluorescent intensities were quantified under a confocal laser microscope. As you can see, the blue fluorescence increased with ND630 concentration-dependent response. The increase in the fluorescence directly correlates to beta-oxidation activity, which turns out that ND630 accelerates FAO activity. Like this slide, FaoBlue can be used to analyze drug effects on beta-oxidation. FaoBlue can also be used for screening drugs related to fatty acid metabolism. Here is analysis of NASH mouse model using FaoBlue. NASH, or non-alcoholic steatohepatitis, is the more severe form of NAFLD, non-alcoholic fatty liver disease. NAFLD is caused by buildup of fat in the liver. When this buildup causes inflammation and damage, it is known as NASH, which leads to scarring of the liver and may lead to cirrhosis or liver cancer. NASH is a typical disease which is characterized by low metabolic activity of fatty acids, and new drug candidates can be screened on the effects of fatty acid metabolism, can be evaluated by NASH mouse models with FaoBlue. The left chart are fluorescence images of mouse hepatocytes isolated from control healthy mice, NASH model mice, and NASH model mice orally administered with a therapeutic agent for hypolipidemia. After hepatocyte isolation from each liver, primary hepatocytes were treated with FaoBlue and fluorescence imaging was performed. Compared with control cells, hepatocytes which derived from NASH model mice showed low fluorescence, indicating low FAO activity. On the other hand, in the NASH model mice subjected to drug, the fluorescence intensity is the same as the control, or rather stronger than the control. This implies FAO activity dramatically recovered by the drug. Like this experiment, FaoBlue is a powerful tool for estimating drug effects and efficiency of FAO activity in animal disease models. The main point I'd like to make in this section is that FaoBlue can be used for various applications, such as comparison to cell types, evaluation of drug stimulation, and analysis of disease model mouse, and so on. Finally, I'd like to talk about two cautions when using FaoBlue. First, the excitation light. It's important to choose proper excitation filters for using this reagent. When using epifluorescence microscope, excited by mercury lamp or xenon lamp can be a bit tricky. Commercial DAPI filters are not compatible with FaoBlue because DAPI filters excite both FaoBlue and coumarin derivative dye. Excitation filters which pass 390 to 450 nanometers are recommended. Please keep in mind that DAPI filters are not proper to use with FaoBlue. The second caution is autofluorescence. When using FaoBlue, please be aware of autofluorescence. Dot-like signals are considered to be intrinsic fluorescence. FaoBlue will often autofluoresce around 400 nanometer wavelength as excitation and fluorescence. We recommend taking data before adding FaoBlue and use it as background when observing fluorescence signals. I'd like to conclude this webinar with a summary. Presently, FaoBlue is the only way to detect FAO activity directly in live cells. The protocol is easy using fluorescent microscope without the need for specialized instruments or technique. Metabolic activity in single cell level can be measured and suitable for high throughput analysis. FaoBlue allows the observational lipid degradation change by drug treatment, which can help drug discovery about lipid metabolisms. Also, FaoBlue has been used in NASH mouse models. Thanks for your time listening to this presentation. I hope you find this overview of our innovative reagent useful in your research. If you have any questions, please feel free to contact us at any time, And for additional information such as downloading manuals, availability, and pricing, please communicate with your regional contact. For Japan, you may contact Funakoshi Company. For the United States and Canada, you may contact Diagnosing. And for all other countries, please contact your local Funakoshi Company distributor.